I'm definitely honored and excited to talk to you guys today. Hopefully, you'll be able to get some takeaways from my journey, which is, definitely a little unique. So I guess just to start things off, let's see. Oops, okay. So I'm sure this is also in the program, but this is kind of an idea of where I actually am right now. So I am currently a graduate student in the UVA Medical Scientist Training Program, which is an MD PhD program. So that means that I've completed 3 out of the 4 years of my medical training, and currently am in the 3rd year of my graduate training. And how I'm actually able to continue my research here at the NIH is through the NIH Graduate Partnership Program, which is allowing me to continue my work with Dr. Jacobson. So, but the question is, of course, like basically, how exactly did I get here? And when I think back to when I was much younger, and even in speaking with my parents, what I started off with in terms of my goals in life, I definitely was not thinking anything remotely close to what I'm doing now. I started off with thinking that I wanted to be a teacher, a doctor, a lawyer, and many times my parents actually had to talk me down from thinking that I could do all 3 at once. So I definitely did not necessarily have a very realistic view of what was possible, but in growing up in my wonderful, I guess, borough of Brooklyn, I definitely had kind of, at least for me, I think a good start to things. So, but when things really started to focus in, or I started to get a better focus for what I wanted to do, was once I reached junior high school. So I attended Andrews Huddy Junior High School, which is one of the, I guess, many specialized junior high schools in the New York City public school system. But at that time, I actually managed to come across this book, which is a book of fiction, so it's a little surprising that this would actually completely change the direction of my life. But after reading this, which is a story about a virus hunter that is tracking down an epidemic, that is the source of an epidemic that's currently occurring, I decided that I was obsessed with all things microscopic. And that was exactly what I wanted to do. I wanted to be an epidemiologist. And in reality, I had no clue what that actually meant, but that was what I was going to do. So this continued through high school, this interest. And in going on to college, I still had this kind of grand idea that I was going to be an epidemiologist at the Centers for Disease Control, and no one could tell me otherwise. So like a lot of my classmates, at least at that time, the vast majority of the students that were entering were pre-med. And so, like all of them, I took my first college-level general chemistry course and was just completely like, okay, well, maybe med school is actually not for me. So I took a step back, and I was still very much interested in infectious disease, but I said, okay, well, maybe I could do this some other way. And I ended up doing a major in human biology with a concentration still in infectious disease and epidemiology. And then again decided, okay, well, now that this is my major, I'll go right back to pre-med. Again, dropped in. It was like, forget about it. And honestly, by the time I graduated, I was adamant that I was not going to do medical school. I felt that there was probably a different path for me. And so despite the fact that I entered undergrad with a very clear idea of what I wanted to do, by the time I graduated, I was very much like, I have no clue what's going to happen. So I decided to take the summer after graduating to kind of get a better grasp of not only what was out there, but maybe what I was capable of. And so what I ended up doing was actually stumbling across, maybe not stumbling across, but I decided to become an EMT, which again was very brief and did not stick. I decided instead that that would be a volunteer position. But even though I was really very much like wobbling and going back and forth, even while in college, I had managed to establish and maintain some really good relationships. And so in speaking with my college advisor, she placed me in touch with Dr. Ann Gershon at the Columbia University. And this was basically as a chance for me to get involved in research. I had been kind of interested in it during my undergrad, but didn't really pursue it. And so in speaking with Dr. Gershon, she felt that I could enter into her lab as a volunteer and basically pick up some of the basic bench research skills that I was lacking while also learning about varicella-zoster virus. So if you don't know, Dr. Ann Gershon is actually responsible for bringing the varicella-zoster vaccine to the United States. So this is her here, and my project with her was actually understanding what components of the virus are responsible for reactivation of latent infection in ganglions. And this was actually a project that also involved working with her husband, Dr. Michael Gershon, who is an expert in the enteric nervous system. So I did this work for about 6 months, and it was really gratifying, and also I gained a lot of skills that I definitely did not have before. And even though it was voluntary, it ended up kind of transitioning me into a lab tech position in his lab. Now, this was a complete 180 because it had nothing to do with viruses. I instead moved on to a project in learning about the pathophysiology of Hirschsprung's disease. Now, this is a pediatric disorder where there is an aganglionosis in certain portions of the gut. So basically what this means is that there are portions of the gut that do not have innervation. And so I was trying to understand what contributed to this, like what cellular signaling molecules are involved for allowing neural crest cells to track along the vagus nerve to those locations in the gut where they need to set up the enteric nervous system. So that was definitely very interesting, and I gained not only a new mentor, but also ended up working with Dr. Eliane Radcliffe. And I mention her because along with the Dr. Gershon, they kind of gave me a better idea of sort of the physician scientist. They were all MDs that were practicing as well as completing research in the lab. And this brought me back to basically saying, okay, well, maybe I should consider medical school again, because I really wanted to do something very similar to what they were doing. And so I applied to medical school, but through kind of, I guess, like what you would call an AMCAS snafu, where they couldn't find some of my coursework, there ended up being a weird process where I never actually was able to complete the secondary applications for that year. And I remember being like kind of bummed, like, oh, okay, well, you know, maybe this just isn't going to work out. But in retrospect, I'm realizing that kind of that whole situation was a blessing in disguise, because instead of just like giving up, I ended up in the lab of Dr. Ai Yamamoto. Now, she was a recent postdoc in the lab of James Rothman, who does a lot with snare proteins, and I guess was a recent Nobel Prize winner. So she was just establishing her lab at that time at Columbia. And honestly, she probably more than anyone else really pushed me in the direction of saying, okay, I think research could potentially be a part of your future career. She definitely loves to remind me now that at the time she wanted me to apply for an MD PhD program. And I said to her face that there was absolutely no way that I would do that because it would take too long. And I'm definitely still in touch with her, and she loves to basically remind me of this. And while working with her, I still kind of transitioned in another direction where I was working on Huntington's disease and other neurodegenerative disorders that involve the accumulation of proteins. And my role was actually understanding the different proteins involved in autophagy and how autophagy could actually be used for potential treatments, and then if there were any other factors that could help in removing some of these mutant proteins. So while in her lab, then, of course, I did end up transitioning to medical school successfully at the University of Virginia. And I kind of tucked away the whole thought of doing a PhD. I just forgot about it. I was just like, well, I don't need that. And instead, what I did was I spent 3 years at UVA, where it was definitely very rewarding, but I still had kind of in the back, like I was really missing research. And I had really discounted it before that it was just a phase, and this is something that I don't need to do, but I kind of had to confront that I really did, I guess, get bitten by the research bug. And so what I did was after 3 years of medical school, I applied to what they call these year-out programs. They're these year-out fellowships. There are a few that are offered through the Howard Hughes Medical Institute, and there was also, I think, one through Doris Duke, and there are several others. But I ended up applying to and being accepted to the HHMI-NIH Research Scholars Program. This, I think, has morphed into the Medical Research Science, the MRSP. But at that time, it allowed medical students from medical, dental, and veterinary students from around the country to come to the NIH and work with a mentor. And I ended up choosing Dr. Jacobson. And this was actually fortuitous because he was working on a disorder that has become really near and dear to my heart. I may not have mentioned, but my family is originally from Jamaica. And he works on human T-cell lymphotropic virus, which at the time I did not know was actually endemic to parts of the Caribbean. And so I was just like, well, here's a disorder that definitely affects my community that I really knew nothing about. Maybe this is a chance for me to actually learn more about it. And it was also fortuitous because I loved the research so much that even after a year, I stayed on a second year. And then once it was time for me to go back to medical school, I was so just kind of upset about having to leave behind the project that I was working on that I was really wondering kind of what to do. And along with people in my lab, and as well as like other people within NINDS, there were several that had spoken to me and said, well, Monique, why aren't you considering a PhD? And again, I kind of had the ready excuses of it's going to take too long, despite the fact that I'd already added an extra 2 years to my medical training, and that it just would not be possible. And I remember actually having a conversation in the hallway on the 5th floor where someone stopped me and said, I want you, like, I don't really want to hear any more of the excuses. If you can actually tell me that when you go to your administration, they tell you no, then okay, that's it. But otherwise, you can't make this excuse anymore. So I did as promised, and I followed through, and I spoke to my administrators. And despite the fact that I thought it was absolutely impossible for me to actually transition into an MD PhD program, they were actually really amenable to this and actually continuing to allow me to do my research at the NIH. So within a month of speaking, well, month to 2 months of speaking to them, they had managed to arrange everything with the NIH administration here. So I guess that's kind of brought me to the point where I can basically speak to you guys. And I guess at this point, I'll just a little transition into what I do. As I mentioned, I work on HTLV-1. This is a human delta retrovirus that was discovered in the 1980s, discovered in 1980 actually prior to HIV. So like HIV, it has a primary tropism for CD4 T cells. However, unlike HIV, it tends to lead to the activation and proliferation of those cells. It doesn't kill them, at least not directly. And there are other immune subsets that are also affected. And what my research focuses on is the fact that tends to be a predominant reservoir in our patients of these infected CD4, CD25 positive T cells. And I'll kind of go on to explain what that means. So it's really interesting that, again, unlike HIV, this is a virus where most of the patients will remain asymptomatic carriers. They will not usually go on to develop any disease. But the two most prominent diseases that they can develop is adult T-cell leukemia, or HTLV-1 associated myelopathy, tropical spastic periparesis, which is a huge mouthful, and I'll just call HAM TSP from now on. And this is the disorder, is it a neurodegenerative disorder that our lab currently studies along with multiple sclerosis. And as you can see, about 10 to 20 million people worldwide are infected. So as I had mentioned, this is endemic to areas of the Caribbean, Japan, South America, Western and Southern Africa, the Middle East, and Melanesia, just to name a few. And so it kind of has an interesting geography. But what you'll actually see in the clinical picture is a progressive muscle weakness, this ataxia, which is basically an imbalance while walking, as well as sort of very stiff lower limbs, very funny sensations that patients speak about in their limbs, as well as sphincter dysfunction and urinary incontinence. It tends not to be fatal, but currently there's no established treatment regimen, so these patients just continue to progress. So as I had mentioned, there is this CD4, CD25 positive subset that are predominantly infected in our patients, and we believe serve as the reservoir. And these cells are actually the regulatory T cell component of your immune system. So there are several different T cells within the immune system, but this particular cell type is important for maintaining peripheral tolerance and immune homeostasis through the downregulation of T cell responses to both self and foreign antigens. And so what this really means, and this is kind of like a nice schematic for what you'd actually see. If you take a normal human T cell and you activate it in culture, it will divide and proliferate, so basically proliferation. And that's what's shown, I don't know if I'll be able to show it here. That's what's shown here. But if you add in a regulatory T cell, you'll get suppression. So basically, there aren't as many divisions that are occurring here. But if you add in a Treg from a patient with HAM TSP, they have a distinct lack of suppression. In fact, it actually looks like there are more divisions that are occurring. And this is something that we know of in the disease, that they have a very activated immune response. So kind of the work that I'm doing right now is understanding what HTLV-1 viral proteins are involved in altering that Treg function. And so I'm kind of focusing on two parts, which is basically how the suppressive capacity is altered through this particular genetic region. And then also understanding whether or not there are these microvesicles or exosomes that are involved. So I hope this is kind of a really complicated, but I hope this picture can break it down. So basically, one of the ways that our genes are regulated is through epigenetics. And what this means is that whether or not there are specific groups attached to different portions of the gene will say whether or not it's turned on or turned off. And it even goes a little bit further about like how stable that expression is. And so this particular region, the Treg-specific demethylation region, is important for Treg function, and it is regulated through epigenetics. And it's not necessarily whether or not it's turned on or off, but whether or not that gene actually is maintained in that on state. And so if you can see here, this is basically expressed here through like the white and black. If it's demethylated, it's a Treg. If it's methylated, it's basically not. And so that's essentially what I've been doing at looking at the different Tregs between our patients and the normal donors. And one of the things that I found was that there's a particular viral gene, HTLV-1 Tax, that if it's expressed in the cells, actually seems to be correlated with a decrease in that demethylation. So I guess as another way to say, it becomes more methylated and therefore the expression of the gene is less stable, which is what we think is contributing to the lack of suppression that's occurring in the patients. But then kind of like transitioning really quickly, I'm also looking at these exosomes, as I mentioned. And these are membrane microvesicles that can contain mRNA, microRNAs, and proteins. And they are we believe now, actively shed from cells. And now it's becoming more known that there are different viruses that are taking advantage of this intercellular form of communication and actually placing viral components in those exosomes. And so I guess I'll just end here that we think that potentially this may be something that's going on in our own patients when we look at the central nervous system. So the cerebral spinal fluid of these patients seem to have these exosomes that contain HTLV-1 proteins. Now, what that potentially means or whether or not it really has any kind of clinical consequence is something that I'm looking into further. So I guess I've said a lot, but just from my own journey, I hope that I can give you some takeaways that, one, don't be afraid to volunteer. No, it's not a paid position, but honestly, that really is what started me off on my journey. And it was probably the most important step. Also, I cannot emphasize enough that I've had amazing mentors along the way. From the Dr. Gershon, Dr. Radcliffe, Dr. Yamamoto, and Dr. Jacobson. They have all been really instrumental in leading me towards this path because they've pushed me beyond my own boundaries and really have pushed me in terms of, I guess, not limiting the expectations that I have both for myself and what I can achieve. And also making sure that you maintain these relationships. I still make sure that I speak to them as frequently as I can and continually update them on my progress. Also, don't be afraid to ask for the impossible. So it might actually seem impossible to you, but perhaps to someone else it actually isn't and is not that big of a deal. So I think a lot of times my own perspective kind of limited me in what I was willing to ask for. And kind of building into that, there was the build up your ego, because a lot of times I just didn't think I could do it, and a lot of it was due to a lot of my own self-doubt. And most of all, have faith in your path. I've definitely taken a more, meandering or perhaps long way to go about things, but I think at the same time it's made me who I am, and I feel like I'm finally on that the path that I am supposed to be on. So yeah, not everyone follows a strict roadmap in life. So I guess I'll finish here.
Does anybody have any questions for Monique? Are you going to be here? Okay. Oh, okay.
This may be a little off. So your picture that you showed of the patient and your clinical symptoms suggested lower extremity problems. Why do you know why the upper extremities are involved?
So for the most part, where a lot of the atrophy that you see for the patients in the spinal cord is within the thoracic spine, but when we actually look at our patients, it's both the cervical and thoracic spine. So it's not really, we don't necessarily understand why it's only the lower extremities. We have some that have upper extremity findings, but it's very few. So it's something that we're still trying to understand. Yeah. But I don't know, Jean, do you want to add anything? Oh, yeah, sure. No, it's going to be from UVA.